Thanks for attending the session for today. The title of the session is Preclinical Studies of Gene Therapy Products, Latest Trends. This presentation will be given by Norbert McCory, the Vice President of Toxicology at Altasciences, and Kellen Koine, who is the Principal Scientist at Altasciences. On the agenda today, one will discuss about our gene therapy experience, actual preclinical experience, and then we'll take a look at the regulatory landscape and study planning. Thirdly, we'll look at what are the most common immunosuppressive regimens that we have seen in the past several years that are given to some of the animals to tamper down the innate immune system. Fourth, we'll then look at biodistribution studies, tissue and vector shedding, and PCR workflow. And fifth, we'll look at then at the assessment of germline integration, because as we know, that's a very critical component of assuring that there is no integration of the gene of interest into the, gonads for the adult animals. So at Altasciences, this is at our preclinical site in Seattle. We have seen a tremendous growth in terms of the number of studies we have conducted over the years. So as you can see from that chart, we have all kinds of delivery, modalities for the genes of interest. We have AAVs, LNPs, and a combination of either and other rare types of gene delivery modalities. So as you can see from there, starting 2021, the earliest year we went, the number of studies was relatively low, speaking in terms of, when you compare to 2024. So as you can see there, in 2024, there was a spike in the number of gene therapy studies for the simple reason that these gene therapy products are really coming into the market, early discovery, now going into preclinical IND enabling, and some of them actually going into even phase 3 clinical trials. And we continue seeing that trend continue up to date, where we are seeing a really increase. In fact, that this trends very well with what we learned at the ASGCT meeting in 2023, that there are over 4,000 products in development, and there are a very high number of posters, presentations, clearly showing that this field is really growing and expanding. So for the AAV subtypes screening in NHPs, this is really important because unfortunately, many of the animals do express antibodies towards the AAV. So as you can see from this table here, which lists the most common adenovirus associated serotypes that are used, the negativity rate is very high. For example, in Cambodian animals, AAV1. When it comes to AAV2, that's also relatively high, meaning that when you are using these modalities, you don't have to screen too many animals. AAV3, AAV5, AAV7, 6, and 7. Now, unfortunately, AAV8, the seronegativity rate is very, very low, as you can see from the chart. And it so happens that this is the modality that is most commonly used currently, which translates to you need very, very many animals to screen in order to obtain the correct number of animals that you need on the study. And as you can see here, we also have been fortunate to compare across different sources. So Cambodian origin, you'll see the negativity rate at only 36%. In Mauritius origin animals, slightly higher, even though the N is lower at 44%. And then when you look at Vietnamese animals, and again, an N slightly higher than Mauritius, but again, unfortunately, the negativity rate there is very similar to the Cambodian, and that just supports the fact that Cambodian, Vietnamese origin animals are really very similar because they come from the same geographical location. And then AAV9, again, slightly better in terms of negativity rate, 68 for Cambodian and 35 for Mauritian animals. And then AAV10 also very good, negativity rate. So again, what this points to is that, especially if you are using AAV8, for as a carrier, it's really critical to really plan way ahead of time to make sure that enough animals are screened in order to obtain adequate number of animals for your study. Okay, so now we'll switch gears and go to looking at very briefly what the regulatory landscape is and what that entails in terms of study planning. So the latest, the guideline that is available is the ICHS 12, Nonclinical Biodistribution Considerations for Gene Therapy Products. Now, this guideline basically acknowledges that products that mediate their effect by the expression, transcription of transferred genetic materials can be looked at slightly differently from the routine small molecule, biologics, oligonucleotides, and things like that. That means that from a compliance point of view, standalone biodistribution studies, actually, it's recommended that they not necessarily be conducted according to GLP regulations. However, it is critical to verify that the data is of quality, integrity, and can be reliable. That being said, it does not mean that you cannot conduct those studies under GLP conditions. It just gives you that option whether you can or not necessarily conduct those studies under GLP. Now, if you are conducting biodistribution evaluation as part of a GLP study, then all the in-life evaluations and sample collection procedures must remain GLP compliant, meaning the QA unit must be involved in ensuring that there is study integrity and the study is being conducted according to the GLP regulations. Now, once the tissues have been collected, sample analysis can be conducted non-GLP. Methods, again, generally are recommended to be qualified, but they can be verified. The most important thing is to really adhere to good scientific practices to make sure that that analysis results can be believed and used for your subsequent IND submission. Now, to continue on the regulatory, and ICHS 12, what does it entail in terms of study design? One, biological relevant species. That is crucial. This ensures that, there's proper support of transfer and expression of the genetic material. Because if you are going to use a species that is not relevant, then that may present problems. Competent tissue tropism, gene transfer efficiency, and transgene expression. That, again, is really important by selecting the right animal species. It so happens that most of the time, the non-human primate will be the most relevant species, even though in a few occasions, the other animals may be relevant. Secondly, biodistribution, the vector or target presence. That means that this has to be shown to be in desired target tissues or biological fluids. But also at the same time, you may be able to demonstrate that it's absent or present in non-target tissues or biological fluids. And then thirdly, you look at the germline. Is there biodistribution of the vector or target itself in the germlines? That's the gonad, ovaries, and sperm. And then finally, safety considerations. What should you be looking at? There's potential for immune response to the capsid proteins themselves. So how do you mitigate that? We'll take a look at in the next couple of slides. There is potential for random integration into host DNA. How do you mitigate that? So biodistribution data coupled with clinical pathology and histopathology to determine correlations with any tissue specific adverse effects. It's really crucial to correlate all those findings from all those data sets to make sure that one is making the proper and correct interpretation of the data. And then there was the ICH-6 that was issued in 2006, which is talked about the general principles for investigating and addressing risks for inadvertent germline integration. And in this guideline, specific considerations that one should take into account recommended was integration of DNA should be minimized in non-target tissues. This is of particular concern for gonadal tissue potential, where there is potential for modifying the germline. Newer gene therapy technologies have resulted in vectors with higher titers, efficient transduction, and broader spectrum, thus increasing the concerns for risk for inadvertent germline integration. Although gene therapy vectors are specifically excluded from the scope of ICH-S6, general principles described in this guideline could be relevant to this product class. And finally, safety considerations, again, as we mentioned earlier on in previous slides. So again, continuing on that in that same breath, if a vector is not detected in gonadal tissue, according to the ICH guideline issued in 2006, there's no further germline integration studies warranted. If a vector is present in gonads, assess whether the level of vector sequence falls below the access limit of detection at later time points, that is transient detection. Now, persistent detection of vector sequences in gonads warrants elucidation of germline transduction. Lipid nanoparticles, or LNPs, may require direct elucidation of gene since there are no vectors. When possible, testing of gonadal tissue is performed to determine whether the vector is present within germ cells or non-germ cells, for example, Sertoli cells, Leydig cells, and leukocytes. So in males, you analyze the sperm at different time points. And again, persistent detection of the vector limited to only non-germline cells around transient detection is in sperm, likely indicates that germline integration has not occurred. In females, if the vector is detected in one oocyte, all oocytes are presumed affected. Lipid nanoparticles can cross the blood-testis barrier. Therefore, again, assess gene of interest when using the LNPs for delivery of the gene of interest. So what does it entail in terms of study planning? Screening of animals is critical, especially for AAV seronegativity if the AAV is the carrier. As mentioned earlier in the earlier slide, especially for AAV8, long-term planning is necessary because the seronegativity rate is very, very low. Plan for the proper immunosuppressive regimen, right? Some instances may require administering of these immunosuppressants so that there is minimal immune response, at least in the first few days of administration of the LNP and or the AAV. Another consideration is animal age and timing of study initiation. Now, there are different study designs. First of all, if there is need for in utero administration, this will need a very long-term planning because you will need sexually mature females, sexually mature males, and then there's a breeding option. And then once these animals are pregnant, you determine at what age you want administration of the gene of interest in utero. So this really requires long-term planning. In addition to the fact that these studies then take, up to 24 months to complete. Juvenile animal studies, again, this is referring to non-human primates strictly. Juvenile animals, again, need long-term planning, 6 to 12 months, to ensure that you obtain animals of appropriate age for administration of the gene of interest, and at what age, is most relevant in terms of expression of the negative effects of the mutated gene or something like that. And then for younger adults, the timelines are pretty reasonable, 8 to 10 weeks of planning. And then for sexually mature animals, again, because these animals are scarce, you need probably 6 months or more to get the animals that of adequate age, but also more importantly, in terms of screening for AAV, especially if you are using the AAV as the carrier. So let's look very briefly at our experience, using the immunosuppression regimen prior to administration of the, either AAV or LNP. Communication and supportive practices in controlling unwanted immune responses to therapeutic transitions of vectors is really critical. Now, challenges beyond precise gene editing include immunogenicity and host reactions, which impact safety and effectiveness. Immunogenicity poses a risk by potentially diminishing therapeutic benefits or triggering immune-related reactions. Host reactions like inflammation or immune rejection can further complicate treatment. There are other reasons, but these are the main reasons why you really need to look very carefully at your product and make sure that there is adequate planning for potential immunosuppression. So there are immunomodulatory drugs that we have used in recent years, and these prevent transgene immunogenicity. They can modulate the inflammation at target sites, and they improve tolerance to gene editing techniques. So these are the common immunomodulatory drugs that we have seen over the past few years. And again, quite a good number of studies do utilize this approach. So the table shown here, it really lists the different types of drugs that we have used. The most commonly used is dexamethasone, and as you can see, the number of studies is quite high. And actually, this data will be updated in the near future because many more studies have been evaluated, using or administering dexamethasone. The next one is prednisolone, and again, the dosage levels can vary, but generally, the dosage levels as shown are the most common. Then we have combinations of prednisolone and diphenhydramine. And then we have other monoclonal antibodies, tocilizumab, tacrolimus, and rituximab, which have all been used to suppress the immune system to assure that there is minimal or no reaction to the administered LNP or AAV. So animals do react differently to the immunomodulators. This is data from only control group animals. And you can see here that some animals showed mild transient increases in some of the cytokines complexes or complement. So on the left, you can see the cytokines there. We saw, in one study, the number of animals, there were reactions. But in the 5 studies, there were no changes in terms of these, cytokines. Again, this is only in control animals. And then on the right side, the biomarker of interest was complement. You can see there was more changes were seen in complement than were seen in the cytokines. So things to take into consideration that there are changes, some of the biomarkers that you could also monitor when you are administering some of these immunomodulatory drugs. Now, we also did take a look at some of the clinical pathology changes and immunomodulators. Lo and behold, we did see that there were, decreases in the counts of lymphocytes, monocytes, neutrophils, eosinophils, and platelets in some of the animals. As you can see on the right side chart, the blue shows an increase in the number of animals that did not show any changes, and there are animals that did show changes in those parameters. Clinical chemistry, we did see decreased levels of albumin, reversible increases in globulin and liver enzymes ALT and AST,
total bilirubin, and creatine kinase.
And in coagulation, we did see elevated fibrinogen, prolonged prothrombin time. So again, really critical to incorporate these parameters in your studies to monitor any changes that could be explained by just the immunomodulators, not necessarily the drug of interest. Okay, with that, I will pass the presentation on to Kaylin, who will talk about our biodistribution studies.
Thanks, Norbert. Now I'm happy to be speaking to you all today, go over some of the points for the biodistribution discussion points. And of course, the in vivo biodistribution assessments are critical for gene therapy compounds, monitor target, non-target distribution, and potential effects, risks, and efficacy. So my parts here are very much from the ground level perspective and what's all involved with carrying out a lot of this work to assess all these gene therapies. For successful biodistribution assessments, you must have successful collections. So think that the work you put in on the front end directly impacts the data on the downstream end. And so for biodistribution, we at Alta commit to clean collections of in vivo samples. So when we use sterile or disposable scalpels, we're going to wipe, we're going to clean when appropriate, and perhaps maybe we order the tissue in the sample collection from the animal if that is going to be the cleanest, or it's going to be maybe there's a risk for contamination of certain organs. So we'll work with you on that. The tried and true sample processing and storage for these tissue collections is flash freezing, so use liquid nitrogen. And we've had much success with our PCR data when obtaining nucleic acids from flash frozen samples. So we also use liquid preservatives when needed. So if we're doing DNA or RNA analysis, or if we have fragile or small tissue collections, but blood preservatives are very important as well, especially if you're doing RNA analyses on those collections. So just something to keep in mind. And at Alta, we have extensive experience with non-human primates, mouse, swine, as well as canines for the range of biodistribution and gene therapy studies. I know Norbert focused a lot on the NHP, so I'll focus on that later on in our discussion as well. And on the right-hand side, you're going to see the list of the tissues that we are highly familiar with at Alta, and also what's driven by the FDA guidance documents, all the documents that Norbert was mentioning earlier. There are the recommended tissues according to the FDA, which are listed here at the top, but this list should be expanded and supplemented if it does not include the expected targets or the relevant tissues, etc. So there are options to expand as needed, but these are kind of the minimum that the FDA is looking for. The biodistribution samples also have additional handling and processing procedures before the data analysis can take place. So before we even quantify the gene therapy, there's a whole bunch of processes that have to be undergone. So we have both column and magnetic bead-based isolation procedures at Alta, and we use those readily. So we're very, very comfortable with them. We have procedures for both DNA and RNA isolations from the whole gamut of tissues, and like the ones that were listed on the slide, but also many other specialty tissues that that particular studies are targeting. And we typically before we isolate all the nucleic acids, we need to thaw and take out an aliquot of the blood, or we have to cut the tissues down to find the appropriate sizes for the lysis procedure, depending on what kit that we're using. For high throughput applications, we have a Kingfisher Flex for use, and we use that for both DNA and RNA isolations. While the magnetic bead-based isolation procedures and the automation platforms were optimized for liquid matrices, such as blood or, say, saliva during the COVID pandemic, as an example, our lab has been successful in isolating DNA and RNA from the whole list of tissue types from the previous slide. So all of those frozen tissue types, we have been able to isolate both DNA and RNA on our high throughput platform, which was generally geared to liquid matrices. So we're very proud of the work that we've done there. We do quantify DNA and RNA from all isolations, and we dilute to the appropriate working concentrations. So nucleic acids are then ready for either qPCR or ddqPCR downstream. Vector shedding. To assess gene therapy target or vector with regard to excretion, they typically urine, feces, or saliva as examples. That's also of interest for gene therapy products in the regulations. Is the compound or target removed via excretion, and how efficiently is it removed? So here, a lot of the biological properties of the vector are considered, perhaps through modeling or details regarding the synthesis process. And is there replication potential for the vector? And what about any molecular variants that it may exist? These are questions to address when planning the vector shedding assessments. A lot of these may be known or answered when kind of diving deeper into the manufacturing process with regard to recombinants or different metabolites as well. And here, since PCR is ultimately the downstream analysis, extra care should be taken during the collections as well. So if we're talking about in vivo studies, it's nearly impossible for any sterile or clean collections to take place of urine and feces, saliva collections. So that's definitely something to keep in mind when you're planning during your study planning phase, when we're talking about NHP or even, rodents and things like that. So this workflow slide applies to the biodistribution. It applies to copy number assessments, vector shedding samples, and it's also going to be applicable to what I'm going to cover next as far as the germline integration goes. So I wanted to give you a better understanding of all of the work that goes into these PCR studies for gene therapies at the ground level. And some of it was repeated, on other slides, but at a high level, once we receive the samples, tissues are cut. They're usually cut frozen. DNA and RNA are isolated, they're quantified, and they're diluted. We do use commercially available isolation kits for both column-based and magnetic bead-based isolations, and we have the ability to handle the wide range of sample sizes, so from a few hundred all the way up to the high throughput scenario where we're doing a couple thousand samples. We also use commercially available master mix kits for qPCR and ddPCR assays. We do have droplet digital PCR capabilities at our lab. We have experience doing mostly singleplex assays, like say a single target on your gene therapy vector, or we also have the ability to incorporate multiplex assays if you're looking for multiple targets, multiple gene editing targets from your therapy. If we're doing reverse transcription PCR with RNA, we have experience performing both 1- and 2-step assays for non-clinical studies. And the data are then, of course, compiled, put into tables and reports, and they can be either standalone or they can be part of a larger tox study. And all of this work here on the left supports all the project types listed on the right. So the custom method development could be PCR primer and probe design. It could be a specialized isolation kit, for example, to transferring an established method from a sponsor and then carrying out a full validation of the assays, which then can be carried through non-clinical sample analysis. So that is our workflow. So for all of this discussion of biodistribution to the organs that were listed on the FDA preferred list, the gonads, the ovaries, and the testes. The S12 guidance that Norbert referred to earlier makes mention of germline integration, so the incorporation of this gene target into the gametes of an organism. So in this case, we're going to oocytes and the sperm cells, right, for the gametes. The first part of this assessment per the guidance is to assess biodistribution. So we perform PCR assessment of the ovarian and the testicular tissues. And so we're either able to quantify the gene target or assessments are below the level of quantitation, so we're effectively zero. If it is determined that the gene target copy numbers in the ovarian or the testicular sample are above a statistically significant threshold, we then will take the analysis a step further to examine the respective gametes. So, and I'm going to go back to the study planning and bring up a note regarding the collections. specifically regarding the ovaries, given their sizes. Ovaries are needed for the other portions of the tox study, right? In addition to biodistribution, say for the clinical pathology, for histopathology. So the collection procedures must be calculated and outlined and addressed in the study planning. Ideally, one ovary is used for tox study analyses, and then the other ovary is stored flash frozen for potential oocyte collection and analysis if needed based on the biodistribution results. For sperm cells, semen will be collected during the in-life at various time points, and samples can be flash frozen until it's determined if gamete or germline analysis is warranted. At Altasciences, we have worked hard to develop and optimize collection procedures for both oocytes and sperm cells using samples from NHP. And we've performed oocyte collections on canine and swine samples as well, as they have become available. To go over the process, from sexually mature females, whole ovaries are collected and flash frozen. But I want to give you a reference. The size of an NHP ovary is about the size of a swollen tick. So that's just to give you a size reference to keep in mind, going back to what we mentioned about study planning and the collections. The frozen ovaries are thawed to room temperature and sliced longitudinally. And then PBS is used to wash the stroma and then release the oocytes in the media. And the oocytes are then collected in tubes, spun down, and they undergo additional washes. And when they are in a dish, they are mechanically denuded of the surrounding cumulus cells by gentle pipetting. Obviously, we want to make basically a single cell solution, but the cells have to be nice and clean and denuded. So the denuded oocytes are collected into clean, sterile tubes with either PBS or lysis buffer, and then they're frozen at -80, so they're flash frozen. From those frozen oocytes, they can be thawed, and DNA and RNA can be isolated. So we had, from our experience, we have had success isolating DNA from the Chi-Amp DNA microkits and the isolating RNA from the RNase microkit in our current repertoire of experience. Some of our initial results. Once the oocytes were collected from frozen NHP, swine, and canine ovaries, and some of these, of note, were frozen at -80 for up to 150 days. So it's pretty cool that we were able to get some pretty good, useful data out of those samples. So each dot on the dot plot on the left side, each point represents the number of oocytes from a single ovary. So you can see that we were able to collect as few as 13 oocytes, and then as many as 125. So the dot plot on the right, then we demonstrate that we were able to successfully isolate total RNA and DNA from the oocyte sample. These are not separated by species on this figure, but RNA yields are in green, DNA are in black. And for RNA, we were able to obtain within the range of 72 to 800 nanograms of total RNA. And then for DNA, we had a tighter range window between 140 and 392 nanograms total. But I also want to speak to the ranges of the data. And, the difference in oocyte numbers that are collected. Each of these collections is dependent on the age of the animal, the estrus cycle stage, species, of course, and the fact that the collections are from a single ovary. They're not going to be from a pair of ovaries because that other ovary has to be used for the tox study analysis. So I feel that those differences are critically important to be aware of. And while we have been able to successfully isolate nucleic acids from the range of these different collections, on a study, there's going to be a wide variety, and there's going to be quite a range of different outcomes, different yields. So just something to keep in mind. Isolation of sperm from semen. This was a very laborious process to investigate and carry out. There's multiple runs, lots of pivot optimization with regards to washing and processing, etc. So from sexually mature male NHPs, the electropenile stimulation method was used to collect semen, which then incubated at room temperature prior to processing for liquefaction. The initial challenge that we faced was once we had a volume of the sample, which will vary for each animal at each collection, how do we best separate the sperm cells from the other epithelial somatic cell fraction? So there was a number of procedures attempted. It's very much a trial and error, old school experimental setup, but we ultimately had success looking into the two methods that were here on that are presented here on the slide, the cryopreservation and the swim up method. So for cryopreservation, an equal volume of cryopreservation buffer was added to the semen sample directly after liquefaction, and the whole sample was then flash frozen in order to preserve the sperm heads, the integrity of the sperm heads. Prior to nucleic acid isolation, the sample is thawed, and there's a series of washes and lysis buffers to counter the cryopreservation buffer. That's all utilized to remove that somatic epithelial cell fraction, which leaves, again, the intact sperm heads, hopefully a nice pure sample to be then used for downstream processing, like isolations. For the swim-up method, which is on the right side of the figure, a sperm preparation medium was added to the room temperature semen sample after liquefaction, which then allows the motile sperm to swim, if you will, to the top of the tube or the sample. So this will form a layer, similar to what you might see in a centrifuge separation. So that motile sperm fraction or layer was then mixed with its own kind of a separated cryopreservation buffer, and then flash frozen for downstream analysis. And for our successes so far, semen was collected from 7 sexually mature NHPs over several days. And the 11 DNA samples reported here on the figure were isolated from and reported for the cryopreservation collection and processing method. And so far, our team has been able to successfully isolate DNA from the cryopreserved sperm fractions. And so that's ultimately what's presented here on this figure. From those samples, the yield range was 120 to just over 2,800 nanograms, which is very successful overall. But what's not included in these DNA results is DNA from sperm having undergone the swim-up method of the isolation procedure, the sperm isolation procedure. The DNA isolation was attempted on those samples, but yields were unusually low compared to what we would expect or what we are used to seeing. So that's why those data are not presented here. But that is an opportunity for optimization going forward. So given what we've learned from our work so far, we definitely have some room for optimization, which is really great. So given our preliminary data, now we have a couple paths forward, which we're very excited about. We've learned such valuable and useful information in all the work that we've done so far, just the past few slides that I showed. And as far as collections go, the oocyte collection is tedious. It requires precision, skill, it requires trained eyes. We're looking into possible preservatives, specifically for RNA preservation, that can prevent degradation and address, say, the speed and the temperature of the collections. You're using a microscope to get these cells, right? So oocytes that are waiting to be denuded and collected sit on a warm petri dish. They're on a microscope. Generally, that's not optimal collection conditions. So we have been involved in optimizing that collection procedure to prevent RNA degradation. So we're excited about those attempts that we'll present at a later time. And in moving towards the PCR assessment of the isolated nucleic acids, we have attempted some PCR using the HPRT gene primers, which were publicly available. So we've performed a 1-step RT-qPCR on the isolated NHP RNA, So we were able to amplify the gene target, and it amplified between cycles like 17 and 34 with an average of 29. So it's quite a range, which we thought was very interesting. But again, nothing to write home about yet. There's definitely room to improve as far as specificity with species-specific primers and also gamete-specific primer targets. So that is where we're moving next. That's, the next step in our journey. And how do we best use our PCR expertise to determine germline integration in oocytes and sperm cells? So in conjunction with all of this, we want to optimize and improve our DNA and our RNA sample yield. The quality of that starting sample affects the downstream analysis and the data at the end. We aim to improve the nucleic acid yields and also improve the quality scores and the ratios to ensure that we maintain that top-notch quality. And that is it for my contribution. So I know Norbert and myself, thank you very much for all of your time and your attention today. We hope you learned a bit more about Altasciences and how we can be of help to you for your next round of non-clinical testing. So it looks like we have a few questions in the chat. So thank you very much to everybody for submitting your questions on today's webinar. First question goes to Norbert. What is the current supply status of non-human primates?
So at Altasciences, we have a very robust supply of animals for use in research. So we have both Southeast Asian origin animals and also Mauritius origin animals. So as I mentioned in one of the slides, the start times generally for younger animals is around 8 weeks time. But if you need older animals where we have to, assess the germline integration and things like that, then that is going to be a little bit longer, up to 6 months, lead time in order to get the animals ready for your study.
Yes, thank you. That goes hand in hand with the study planning slides that we were discussing. The next question is related. We've talked about kind of planning ahead and making sure we have the appropriate aged animals. So what are the typical lead times to initiate a study in NHP, in monkeys?
Right. So as mentioned earlier, so that approximately, for young aged animals, that would be about 8 weeks. But if you need much younger animals, juveniles, that may take up to 6 to 12 months. And for older animals, about 6 months to get the study initiated.
Thank you. I think we have maybe just time for this one last question. Do you have any historical background data for inclusion in reports if needed?
Yes, so at Altasciences, we do correlate all, data from control animals and put that together for utility, just in case there is a case to argue against a finding, which we know is a normal background finding. So yes, there is availability of historical background data that can be used on final reports.
Thanks. So thank you very much. It was great to speak to everybody today. And with that, I think I'll turn it over to our coordinators, who will be happy to send us forward on any questions or feedback that you all have. Thank you very much for attending today.